Hi everyone, welcome back to my channel Pharmagist. Myself Dr. Haimavati. In today's video we will be discussing about AlphaFold. So I have kept the title AlphaFold. Is it the end of homology modeling tools? Why is it so? We will be discussing in the coming slides. So AlphaFold is an artificial intelligence system which is developed by DeepMind and EMBL, European Bioinformatics Institute. So AlphaFold, it predicts the protein structure from the amino acid sequence. This was launched in 2021. So it is a collaboration between EMBL's EBI and DeepMind software company. So both these together, they have introduced this AlphaFold. They have tested it well. Even the CASP14, it has shown a benchmark in predicting the accurate structures. And this AlphaFold, it is nothing but a homology modeling tool which is developed by artificial intelligence systems. So in this, the role of EBI, that is European Bioinformatics Institute, is it curates and organizes the data set of this AlphaFold, and then it links it to other biological data resources like PDB and UniProt. So it covers the sequences, entire sequences in the UniProt database. Here DeepMind is a scientific discovery company. So EMBL EBI, so it is a part of European Molecular Biological Laboratory, and EBI is European Bioinformatics Institute. So this UniProt, it has over 200 million protein structure predictions which are given by AlphaFold, and these structures, they are freely and openly available to the global scientific community. So when you see the website of this AlphaFold, so this is the website of AlphaFold. So it is a protein structure database. So here you can search for this protein, gene, or UniProt accession number, or you can give the organism also. So they have given the examples also here, like if you want to search for a protein like a free fatty acid receptor, or if you want to search a gene, you can search it by the gene name, or if you have the UniProt accession ID, then also you can search in this database. Or if you want to search for the sequences of the E. coli, then also you can give the name of the organism, and you can search for different protein structures. So generally in homology modeling, what we do, we have a number of steps. Like we search for the sequence, then we align it using the BLAST, we search for a template. After getting the template, we align both the sequences, and then we submit it to homology modeling software like Modeler, and then Modeler generates a structure for us. After generating the structure, what we do, we have to validate it using the PROSA or errat, etc., etc. So there are a number of steps involved. But in AlphaFold, so the best thing here is we need not do all these steps. So it directly gives away the structure of the organism. It directly gives away the structure of the protein. So how this happens, I will show you. For example, if we have a UniProt protein ID, so for example, I have searched for a protein which is not having any crystal structure. So we develop homology models for proteins which do not have any crystal structure. So here I have searched for a protein structure. So this is a ubiquitin conjugating enzyme E2V2. So its UniProt ID is G3V113. So we will give this UniProt ID here, G3V113, and then here human. We will search for it. So here you can see it has quickly given the result. So you have got this. So now you can click on this. After you click on this, so here it has generated the PDB file, the MMCF file, and the predicted aligned error. If you want, you can give the feedback on the structure, looks great or could be improved. So here you can see it is giving the information on this protein. So we have given only the UniProt ID here. So when you have given the UniProt ID, what the results are, you got the protein, gene, the source organism, and see experimental structures, none available in PDB. So there are no experimental structures or no PDB structure available in the protein data bank. Hence, because there is no protein structure, we want to develop a homology model. So this homology model here, see, it has been given by this AlphaFold protein. So you got the PDB structure. So just now I told no that we have different number of steps involved in our generation of the protein. So generation of this homology model of the protein. But AlphaFold, what it has done, it has made our life easy. So now directly you can give the code of the protein or name of the protein, and you can get the PDB file. So when you click on this PDB file, it will give you a PDB file. So you can just download it, and I have downloaded that, and you can open it in any visualization software. So just now it is downloaded. Just a moment. So what I mean to say is by using AlphaFold, so all the steps that are involved in homology modeling, so all those are overcome. Here you can directly give the protein structure. So you can directly search for the protein, gene, or give the accession number, and you will get your protein. So here is our protein. So you can see, so this I have opened it in Discovery Studio. Here you can see the protein. We can change the background color so that it is visible more clearly. So see here, this is the protein that is predicted by AlphaFold protein database. So this is the protein. So now this protein, you can use it for your study, that is structure based drug design or any other design, inhibitor design. So whatever study or scientific study you want to make, you can use this protein structure. So in this AlphaFold protein structure database, so this is the best part. So you need not search for BLAST, you need not align it, you need not submit it to a modeler, and again after that validating is also not necessary, because it predicts very accurately the structure, so that we can use it in our research. But sometimes you should be careful also. If you want, we can validate the structure, and I have validated the structure, and it has passed or qualified all the prosa plot and error plot. So here they have given the success stories also of AlphaFold. So millions of researchers are using this AlphaFold in their research. So here it has been used in malaria drug discovery, in fighting osteoporosis. So there are a number of case studies showing the use of AlphaFold. So AlphaFold unlocks one of the greatest puzzle in biology. So it helps researchers piece together one of the largest molecular structure in human cells. So I encourage you to go through this AlphaFold database and read through it and apply it in your own research. so that now homology modeling will become a matter of the past. That is why I said is homology modeling tools. So AlphaFold has put an end to all the modeling tools and validating tools because it predicts really accurate structure. But one more thing is, whenever we are using artificial intelligence, because it is based on the data that we provide, so this figure also should be kept in mind. That means this should be taken with a pinch of salt. Sometimes artificial intelligence may predict the wrong answers also. That means we see it as a tiger, but when you zoom it and see, it is not a tiger, it is a dog. So such predictions may also be possible. So there is possibility for such things also, so that we should be careful. So whatever the output is generated by AlphaFold or any other artificial intelligence system, we should not blindly take it, consider it and go forward. We should check it. So we should submit the data, we should use artificial intelligence, and no doubt about it, we get good results. But once we after getting the results, we have to check them carefully whether they are correct or not. After careful checking, then we can proceed forward for our work. So checking at the initial stages is the best thing. So whenever you're getting the output, check for the output whether there are any errors or not, and then you can proceed further and continue your research. So that is about today's video. I hope you liked it. So we'll see in the next one.